that they were working with a bacteria called Streptococcus thermophilus, which is used for making yogurt in the industry. So they wanted to improve the strain that they had in the company, and they were experimenting with different bacteriophages. So what they found out that if when you basically infected these cells with some bacteriophages and they recovered, and you did the infection again, second time, they were way more resistant to the bacteria, to the bacteriophages. And looking into the reasons for that, they found out that there is a particular genetic locus in this bacteria, which actually acquires a lot of the viral genetic code and stores it as in a library for future reference. So every time, the next time this particular virus comes along, it recognizes the sequence from this virus and then unleashes the Cas9 system, which chops up the DNA, and that's the end of the viral infection. Very ingenious, fantastic way of antiviral protection. So how did this particular paper fuel the boom of CRISPR-Cas9? So this particular slide of mine would guide you through how it actually works. So can you guys see my cursor moving on the slide?
Yes.
Okay, great. Thank you. So this is what is known as a CRISPR array, and this is present in the bacteria. So these green portions are called protospaces, which are nothing but the sequences which were there in the virus, and it has been chopped off into small pieces by the bacterial RNA bacteria and put it into its own genome. So this is a sort of a locus. What happens is so these are transcribed as the pre-CRISPR RNA, and then there is a combination of RNase 3, and RNase 3 actually chops them up into smaller bits. As you can see, this is called pre-CRISPR RNA processing. So this processing leads in the last stage to a complex which has this blue Cas9 protein, which is the endonuclease. It also has something called a transactive scaffold, which helps the protein to change its conformation and be active. And also the small green bit, which actually takes the whole complex, takes the scissor, if you may, the Cas9 scissor, to the place where it's supposed to cut. And there are many of these complexes because there are so many viruses the bacteria has encountered over the years. So in the next step, what it does, it goes to a genomic sequence, if it can be viral, or in our case, we have modified it to be our own genome of interest of any species. So what they do is basically, this is the target sequence already present in the host genome. It matches up like this. It matches up the green portion along with this green portion. Oops, I'm so sorry. And then it also recognizes this purple, this is called the protospacer sequence. And then this is a very important piece of sequence which this particular protein recognizes. Otherwise, it might go and bind a lot of other places where it's not supposed to. And then what it's done is basically the DNA scissor actually works, and it does a blunt end DNA cleavage in both the strands, which leaves the DNA exposed, and the other endonucleases in the cell clears it off. So this is a fantastic way where how bacteria actually uses the system to take care of the viral infections. Now, how have the researchers used to do this? Now, what the researchers saw that you have this green bit, which you can actually change. And if you change this, And then the whole complex can actually target any sequence that you wish in any genome. That's a fantastic thing. So, what they did was they synthetically, mind you, this is all done synthetically. They had a sequence, let's say you have an insulin gene which you want to delete. You would probably, oh, what's happening? You would probably have the insulin gene sequence. It has to be a certain number of nucleotides, generally around 20 nucleotides long, and you would certainly not get the entire gene, but you would be able to disrupt only a part of it. And when you disrupt a part of it, the idea is the whole gene is basically, or the protein that it produces is a missense protein. It's an unfolded protein, and it's getting chopped off. So that's how you get no expression of the protein. So you synthetically design a sort of a segment of like a PCR primer, then attach it to a scaffold, which is important for activating the Cas9 protein here in the blue, and then you mix them both together inside the cell. Now, there are several ways of delivering this particular complex into a eukaryotic cell. There are plasmid-based options where you actually express this entire RNA and also the protein inside the cell using eukaryotic promoters to produce this. You can also make them through lentiviral approaches. So basically, you pack these things into a lentiviral genome and then infect or use the plasmids having this lentiviral packaging material into the cells, and they express these things because the lentiviral genes have all the mechanisms to express exogenous protein into the cells. And also, you can have this particular protein purified, no viruses required, have this synthetically synthesized, and then put them together on the lab bench and then electroporate them inside the cell. It doesn't matter how you actually get the protein and the RNA inside the cell. Once it gets there, it does what it's supposed to do. So it forms the complex like this, as it's supposed to, the scaffold helping the protein to activate, and the sequence is interacting with the identical sequence in the host genome, recognizing the protospacer motif, and then it does the double-stranded cleavage. So this is pretty simple, and that this all happens inside the cell. You have no control over how this one happens. After this, what is interesting is the cell, all mammalian cells or all eukaryotic cells have some kind of DNA repair mechanism. It is the most commonly used two are called the non-homologous end joining method, and the other one is homology-directed repair method. So what happens in the non-homologous end joining is basically if there is a nick, this mechanism tries to repair it, and this repair process is extremely error prone. So what it does, it by mistake, maybe, oh, I'm so sorry. Right there. By mistake, what it does is it adds on, let's say, a couple of nucleotides, or it deletes a couple of nucleotides from there, or there is, because of this insertions or deletions, the whole thing is frame shifted. So basically, all three of these would result in a non-functioning protein when it's translated. So that's how, and then you would get no activity for the protein, and sometimes if you're lucky, you'll get no expression of the protein because the mRNA is so messed up, it'll just not pass the quality control inside the cell for the RNA. So this is how we get a non-functional protein using CRISPR-Cas9. The other method, which is a more subtle way of dealing with things, is basically you don't want the entire gene to be destroyed. All you need is basically to have a single or a couple of changes in the genetic reading frame. So what you do is you basically provide the cell a repair template, a template where you have made the changes. So this is the break that we got from this particular step. And then you have this particular repair template where you have this orange bit is the change that you have introduced and you want it to be copied into the genome and stay there for perpetuity. So the cell actually takes this help from this repair template and repairs this gap by reading this change. So this change is then incorporated into the genome for perpetuity, and it gets passed on to your other generations as well. So this is how you get a small but subtle change in your genome. And if you want a bigger change, you go for this non-homologous end joining process. So these are the two main methods CRISPR-Cas9 actually works. Um, and people have used this, and as I'll show you later on, they have modified and engineered this entire process to do all sorts of things. But starting the base of all genome engineering using CRISPR-Cas9 is this particular process. Okay, so now coming to the second part of the talk is how is CRISPR done in the labs? So I remember reading PCR back in my bachelor's days, and I was fascinated about polymerases and everything. But the very simple thing that was not taught to us very clearly was the most important thing in a PCR is the primer. Because without the primer, without the designing of the primer, you cannot run the PCR. So I would try to demystify how things work in the lab a bit. And the way it is done is basically uh, you have to have a gene of interest in your mind. Let's say you have a gene which regulates a process of let's say amino acid metabolism. So you want a particular gene A to be deleted, or at least have portions of it deleted. So what you do, you go to your genomic databases, which are plentiful for humans and other species, and you select a portion of your uh, DNA where you want to target. And there are lots and lots of predictive websites, algorithms around in the internet, which would give you the, if you put in your sorts of 2000 base pair sequence, it will suggest good good spots for designing the guide RNA. The guide RNA, in the sense, as the name suggests, it guides the Cas9 protein towards its activity site. So you can design it.
ಇಲ್ಲ ಅದು
You
ಇದು
can also,
ಮುಂಚಿಂದ ಜಾಯ್ನ್ ಆಗಿ ಅಂತ ಕೊಟ್ಟಿದ್ರು ಅದು ಆಡಿದ್ದೀನಿ ನಾ ಆಫ್ ಆಗಿದೆ
uh,
ಇಲ್ಲಿ ಏನ್ ಮಾತಾಡ್ತಿರೋ ಅವ್ರು ಕೇಳಿಸಲ್ಲ
right. Can you hear me? Yeah. Okay. Right. So the guide RNA is now designed with the help of the predictive algorithms in the from the internet, and there are plenty of them. And then you can order them from different commercial companies. Many of them now in India. It's a very nice thing. And then you have to introduce these guide RNAs along with the Cas9 protein. So the Cas9 protein is generally produced in bulk quantities in E. coli and then purified, standard procedure. Um, and they have to be certain quality to be put into a eukaryotic cell, because otherwise there would be a reaction, an immune reaction, and the cells would die. So you need to be careful of that. You complex these things into a Cas9 guide RNA and protein complex, and you electroporate them. You do it plasmid based, whatever you want to do inside the cell. Now, you have a pool of cells which you have electroporated. You would not imagine that all of the cells would have undergone or would have undertaken the uptake of this complex. So you have to basically, you have a pool where you have some cells which have undergone electroporation, some cells which have not. And how many cells have actually taken up your sort of cargo of interest would suggest that how much of your efficiency, how efficient is your process. So you basically go ahead and do some NGS, next generation sequencing, on your pools to find out the efficiency of this particular process that you just did. And after, if you're happy with the efficiency, if it's more than 50, 60, 70% of your pooled cells have taken up the complex, the cargo, then you go ahead and do a very labor intensive process of called serial dilution or serial dilution cloning. The idea here is basically to start with a flask full of cells, of which many of them, the blue ones here, have taken up your cargo. Those are of your cells of interest, and then put them sort of there's a formula that if you put a certain dilution of this particular cell or solution, you would theoretically have one cell per well of a 96 well plate. So that's in theory. So you follow that maths and you know exactly how much ml or microliters of stuff you have to put into each well, and you do that, and you grow this particular plate for a while. After a while, you take those cells when they have grown up quite a lot. You go back and you do this next generation sequencing on the entire plate with 96 samples, and that's why next generation sequencing is so good, because it's extremely high throughput. So now, from the results of this sequencing, you would know that which particular wells have the sort of the cell line which had actually taken up and done the job that you wanted it to do. So you go back to the plate and you pick the particular clones, like picking clones from a petri dish in a bacterial cell. For the bacteria, you pick those colonies from the plate and you expand them and you stock them up. And this is your cell line. This is your modified cell line of interest. And if you want to double check that, you do a Sanger sequencing on the locus that you wanted to change, and you basically have a fully functional cell line that you want the gene to be deleted. And then, of course, there are other downstream processes that you confirm phenotypically that these particular cells do exactly as it's supposed to after the deletion, as you would check in any sort of mutation experiment. Right. So I have put forward a timeline to just to suggest that how time consuming this process is. So as you can see, it is from the first time you sort of put in your cargo of the Cas9 and the CRISPR, it takes around 2 months to get to a stage where you can actually start using your reagent, your cell line, for downstream experiments. And you, of course, so in a lab like ours, where we have multiple genes to delete and modify, we run at least 10 genes in parallel for do this kind of large scale uh, deletion assays. There are other ways to do this as well. People not only have a target in mind sometimes, people want to do a genome wide screen. So we don't know for a particular thing, let's say we don't know uh, what are the particular genes that are involved in the process. So what would you do? In the bacteria, you would go for a transposon mutagenesis or saturated mutagenesis study. In here, you do a CRISPR screen. You basically have guide RNAs, which would take the Cas9 for the cleavage to all possible genes in the particular cell. And then you select, after you have done all this process, you select for the clones, which, uh, or let's say for a drug treatment, you want to sell and you want to know what are the genes responsible for providing resistance to this particular drug. So you do that after the CRISPR screen, you select the screen against this drug treatment, and then you have a list of genes which you think are important in drug metabolism. So it has an extremely wide application in pharmaceutical industry. Now people are going ahead with the CRISPR screens left and right in the industry and in academia to understand the genetic network. And it's a very hot field. It's a very upcoming field. So I would suggest if some people are interested, they can actually start reading up on these. And this is a fantastic field to be working on. Okay, so how do you analyze these experimental outcomes? So the analysis bit is basically to understand which of those 96 clones. It can be 96 and more. It depends upon how you handle it and how efficient is your process. So, how do you actually know which particular sort of cell has undergone the CRISPR process, the gene editing process? So, there are basically three different types of things that you can do. The very easy and the most crudest form and the easiest is to do a T7 endonuclease assay. The T7 endonuclease is the endonuclease which basically cuts the DNA whenever there is a mismatch. So in CRISPR, what we are creating is basically a change in the genome. And the idea behind this assay is to have uh, and also not all of our cells would be, so not 100%, our process is not 100% efficient. So there will be some cells which are still wild type. So what this process does is basically PCR amplifies the region of interest where you're supposed to have the change. Then you basically melt them and re-anneal them slowly. This re-annealing process actually makes what makes it interesting, because you when the different parts starts to re-anneal, there is a possibility that a wild type strand will anneal with a mutated strand. That way, you will have a mismatch in the final double helix. And if that is present, you introduce your T7 endonuclease into the mix and it will make a cut. So if you run after doing this assay, if you run this particular mixture on an agarose gel, you would see multiple bands, and those multiple bands would suggest that there had been some different kinds of combinations of changes that has happened in your gene of or region of interest, and your wild type should be one fat single band. So that's how you get to know that something has happened. But the problem is, this is not quantitative at all. This is purely qualitative. Low throughput, you can't run more than, let's say, 30 at the time. It's also difficult that way. But it's fast and it's very easy and it's very cheap as well. So the next best thing you can do is basically you have Sanger sequencing. You do Sanger sequencing all the time. It's very standard procedure to sequence DNA. So I'm not going into the details of that. This is fast and also easy. It's semi-quantitative. It will give you some hint about what's going on, but it's medium throughput. It's not high throughput, and it's analysis software dependent. So you can't do it manually anymore. You can't run a gel to see. But the gold standard for this analysis is the next generation sequencing. It is fast, no doubt, but it is difficult because you have to make a library out of your 96 clones, which is time consuming. It is expensive. Yes, it is very expensive because the reagents are proprietary from a particular company. You can't make it in the lab. But it's very quantitative. It will give you exact exact amount of changes, a number of changes in the entire process. So out of 96, it would tell you that 10 of your have this sequence and this is the change. Fantastic. It's also high throughput. You can actually run up to 384 clones at the same time if you want to. But it's also analysis software dependent, and this analysis a bit more sort of advanced than the Sanger sequencing. But nonetheless, they are now all standardized. If you want, there are lots of tools available on the internet, and any sort of bioinformatician would be able to help you with this analysis of this particular thing. So that's the best thing. That's how you analyze your CRISPR applications. Now, coming to the part where we have done all those kinds of CRISPR treatments, but what are they used for? What are the applications for this technology in the wider scientific world? So the first is, of course, a typical question a scientist asks is genetic deletion to study the function of a gene. And that way you can do by creating insertions and deletions, which is called indels here. So you have a gene of interest and you do CRISPR, and then you get a deletion or insertion in the gene of interest, and that sort of destroys the function. You can also do insertions or replacements. So this is very useful if you want to introduce certain sort of genetic mutations. So let's say there are lots of genetic single nucleotide polymorphisms present in the human genome associated with human diseases. Very important sickle cell anemia in hemoglobin. So if you want to study those in a cell line, all you need to do is use a template which has the change, and then you have a cell line mimicking the exact sickle cell anemia in a cell line disease model, which is fantastic. You don't need a big host anymore. You can do it in the cells. You can have very large deletions. Let's say you want to delete two genes of eukaryotic life, can be easily into the tens of kbs, kilobases long. All you do is have to have two different guide RNAs at both the ends, and hopefully there would be a large deletion that actually gets rid of the entire genomic loci. So then you have something called a gene activation and gene sort of switching on and switching off of DNA that can be achieved by just having the nucleus present there. This is very important for epigenetic studies. Also, you have, sorry, this is the genetic thing, and this is important in for having to study the transcription biology of the gene. You can have activations switched on, switch off of the genes. Also, it's very important now, over the time people have used it for uh, microscopy, because Cas9 protein is extremely specific for the genomic sequence that guides it. And if you have a particular locus to visualize, then all you do is tag a GFP protein along with the Cas9, and you put it and you use a guide RNA to target it to a particular genomic loci, and then you'll see it under the microscope glowing like a Christmas tree. So these are some of the many applications of the CRISPR technology. Some of them has been further modified for changes in crop, changes in animal productivity, drug resistance, and all sorts of stuff. So clearly, it has a lot of potential. Okay, so this is the last part now, where I would sort of give you a hint about how I use the CRISPR-Cas9 in my system, my research, and also what are the future trends that we can actually think of using it. And some of the future trends, I think many of you who are young and supposed to join the scientific world would be working on when you actually get to the lab and you do research, the cutting edge research. So I'm very hopeful of that. So I basically work on host pathogen interactions. By that I mean bacteria and immune cells. As you know, immune cells are the first, or innate immune cells are the first line of defense against any infection. So when a bacteria invades your body, this particular cells, uh, the macrophages are the first line of defense. So I study how they react to bacterial infections. So but there has so when a cell is attacked by a bacteria, there are multiple options available to deal with this bacteria. One of them is cell death. So a cell undergoes suicide to stop the bacteria from infecting any further. And also, the cell suicide process actually helps to raise alarm to sort of alert other people, so other cells surrounding, that see, I'm infected and there is a problem. So can you please come and help me? So this is how actually in TB, the Mycobacterium tuberculosis, gets encapsulated into a necrotic foci, where it has been contained by the macrophages, which are constantly trying to keep it under check. There are various processes of cell death. Three of the main ones are called pyroptosis, necroptosis, and apoptosis. You are most familiar with the word, I guess, of apoptosis, but apart from that as well, there are a couple of others recently discovered in the last couple of decades of different ways of cell death. So this is the main, broadly, I work on these factors. So going into a little more detail is I work with non-model species, the chicken species, the chickens rather, and it's an avian, it's a bird, it's an avian species, and as we have heard a lot around the world, that chickens are generally uh, full with salmonella. So their meat and their eggs, if they're not treated properly, salmonella infection is a part of chicken consumption, especially in the developed worlds. Non-typhoidal uh, salmonellosis is a big problem, and it's a problem in animal husbandry, in chicken rearing. So people are looking at different ways of vaccinating the chicken against salmonella infection. And as you all know, if we have to develop a vaccine, first we have to understand the pathogen, how it reacts with the host cell. That's exactly what scientists are doing all over the world now against the virus that we have around with us. So, Salmonella typhimurium is a zoonotic infection that is, and its pathogen, but its pathogen is very commensal. It doesn't harm the chickens at all. That's how the chickens are a good reservoir for this bacteria. But once you consume this infected chicken, generally not properly cooked, and that's why it's a problem mostly prevalent in the Western world rather than in the eastern parts of the world because we cook our food a lot well. So there are certain proteins called the pattern recognition receptors, which immediately recognize this particular bacteria, and they unleash all the sort of arsenal that they have. But there is a difference between the immune sort of cells or the genes that a chicken has compared to us. So the humans and chickens vary quite a lot uh, in response to this, as we expected, because they are birds and we are humans. But bird seems to be immune to this. So why? The question is why. And those are the two questions that drive my research: is why does the mammalian, ours, and avian innate immune system have completely different responses to the same bacteria? Are they seen differently? And what are the molecular responses of the chicken innate immune cells when they see salmonella? So if you understand this, you will understand how to sort of intervene and stop that from happening. For doing that, so this is a sort of a cartoon in comparison of mouse, which is sort of the proxy for humans because you can't really work with humans that easily, and the chicken ones. So I'll guide you through this a bit. So these green dots are LPS, lipopolysaccharides, extremely present, very high prevalence on bacterial surface, and these are recognized by, sorry, these are recognized by something called a toll-like receptor. These are all pattern recognition receptors present on the cell surface, and they initiate a signal transduction, and that activates IL-1 beta. This is a cytokine. This is interleukin 1 beta production. Second, this is a bacteria, and these are all flagella coming out. That is recognized by, and this flagella is injected into the cell by the type 3 secretion system of the bacteria. So bacteria has a needle-like structure which puts in certain kinds of protein into the host cell. Once they are in, they are recognized by another cytosolic protein called the NLRC4, again a pattern recognition receptor, and that goes through a caspase 1 activation protein. And the important bit is this gasdermin D protein, which actually forms pores in the cell membrane. Now, these pores are very important because of this pores are formed, the cell actually dies because you leak out all of your stuff through these pores, and eventually the cell dies. But also through their death, the signal through this IL-1 beta, which is a pro-inflammatory molecule, it signals to other cells, other immune cells, that this is a spot which needs attention. So please come and help. So neutrophils come into their help. Now, if you look closely into the chicken ones, here, unfortunately, you don't have many of the components that are there. Like vitamin D is absent in chickens. There's nothing of that sort in the chicken genome. You don't have NLRC4 in the chicken genome. So how does this happen then? So we actually have big question marks in after this process. So after this bug actually injects flagellin inside the cell, what happens is completely unknown. So it was my task to understand what is happening, which pathway is being activated to get the cell to die, because cell death is very, very important and fundamental to all our immune responses. So I'll cut the very long story short, and what we got at the end, that we saw that chicken cells die in a different way than from mammalian mouse cells. So they are different, and the way the difference is, there's a classical method of cell death. Excuse me. There's a classical method of cell death in mammals, as seen in mouse and humans, but that particular pathway, which is called the pyroptotic pathway, is not involved in chicken cell death. So how do they die actually? So it's a very complicated picture. Forget about everything else. Just remember this RIPK1 and RIPK3 and MLKL axis. So this is the third way of dying. It is called necroptosis, and this is generally not used for cell death purposes in mammals, but we will see how this is important in chickens. So my focus was this RIPK1, RIPK3, and MLKL proteins. And MLKL also forms spores, which helps the cell to die. Okay, so this is the pathway in a much more simplified form. You have this death receptor getting the signal from upstream, like the detection of the bacteria. Then the message passes through three particular proteins called RIPK1, RIPK3, and MLKL. Oops. Oh, God. Yeah. Uh, so RIPK3 is the main signal transducer. So the RIPK1 is one of the signaling molecules. There are others which come and directly interact with RIPK3. So the RIPK3 is a hub protein which interacts with the death process. And MLKL is the effector molecule which actually forms the pores. So these two are the most important proteins that you could target in this process. Now, the way science works in the labs is basically you theorize, you have a hypothesis that this might be the case. And how do you prove that this might be the case is basically you knock these genes out, you delete these proteins, you stop their activity, and then you do the same thing, same assays to see if that made any difference to the process. If it makes a difference, then yes, this is different. This is important for that particular process. And that's how you came to, this is how the results are. So this is some kind of a inhibitory assay. So basically what I did, I had a chemical inhibitor which stops the activity of RIPK3. And as you can see that I had added this inhibitor in different concentrations in 0.25, 0.5, 2 micromolars. And this is the untreated sample. And compared to this untreated sample, you see a reduction in the percentage of cell death when they are treated with salmonella. So the wild type dies 80%, my treated samples die less, 60, 50%. So that means this particular molecule has some effect. Now, given this, I was sort of encouraged to go on to do CRISPR sort of deletion of this molecule. This is what you sort of get as a primary transcript, like of the RIPK3 from chicken. And there's a lot of sort of optimization that is required as to where to target your CRISPR-Cas9 system. So you generally target towards the 5' end of the protein. So once you do that, you go through the whole process of generating CRISPR, as I mentioned earlier. And then at the end, you get this 96-well analysis from the next generation sequencing machine that tells you that which well. So each circle, each pie chart is a colony, is one colony of cells. And they will suggest that which colony is a knockout by highlighting this KO here. So you can get multiple knockouts from these particular cells, and you screen these mutants to form, oh, what's that? Yeah, oh, sorry about this scribble. And you screen these particular clones for your activity. So Here you would see that clone number 22 and 24, don't worry about where they are. They were almost at the same level where when I used my inhibitor. So this is the wild type untreated sample. It dies 80% when salmonella interacts with it. But when I delete these particular gene, my cell death drops to 40%, like 50% reduction, which is which is suggestive of something. So in this way, when I have deleted the function of this RIPK3 protein using CRISPR, I could see basically a phenotype of that particular molecule, and this will suggest that this molecule is important for this process of cell death. Now, again, I have to prove it once more because I have, and science actually is built upon multiple um, proof of the same thing. So it's just not enough for having one example or one particular gene. Somebody might say it was just by chance. So you have to prove doing this. What do you do? Uh, Somebody is using the pen on the screen. Anyways, so what is that is now is the turn of the MLKL protein, which is the effector molecule. And I did the same thing, went ahead with the transcript and I deleted it. But now you would see that instead of 96, I have only 37 clones. That's because you not there are good days and there are bad days, and this is the bad example. But still, you can't really help. You only get two or three knockouts out of this. You have to work with that. And by the way, I should also demystify science because science is not a straight line. It's not like going straight from A to B. It's A to B through all sorts of intermediates, all sorts of failures. So be prepared for that. So you get those kind of knockouts and you do a assay, again a cell death assay, how much of your cell dies when they see salmonella, and you see this is the untreated cells and this is my knockout. And there's a significant drop, and this drop is statistically significant. Multiple repeats, 4 or 5 times I've done this experiment. So they're all important. So this is how you actually do science. This is how you use CRISPR-Cas9 as a tool to deal with your question. So, okay, this is whether this gene is important or not. Previously, without this particular CRISPR-Cas9 system, I had to generate a mouse with MLKL deletion. It's called a transgenic mice. And the amount of time it takes and the money it takes to generate a transgenic mice is humongous. It's huge, and you have to wait for at least a couple of years to get that thing up and running. If you're lucky, this mice should have already it's already somebody had made it. If you are not, then you are stuck. But using CRISPR-Cas9, you basically cut down your 2 year timeline to 2 months or 3 months. And that is a big boost to the productivity of the scientific community. So that's why the CRISPR-Cas9 is so popular and people are going ahead with that and sort of modifying it with such a passion. So that's the end of the CRISPR story as of my usage. And but what is next? Well, how do I even develop this even further? So, what is you remember this slide that what are the two main ways of doing CRISPR repair? Let's focus on this particular subtle changes, like one or two nucleotide changes. What I mentioned early on is basically having a template with your change in there. But this process is extremely inefficient. Only 1% efficiency rate. Out of 100 tries, one try of yours would be successful. Completely unacceptable. So people have tried to increase the sort of efficiency of this. And how they have done it is something called a base editor. Now, you all know so DNA is made up of nucleotide bases called ATGC, adenine, cytosine, uracil, all sorts of those things, ATGC, guanosine. So now what you do is basically, if you can change a G to a C or G to a T, then you have your change. You don't need to do any sorts of other things. But how would you do that? So the people, very smart and clever people, came out with the idea that why not tag two proteins together? One is Cas9, Another one is a cytidine deaminase. So if you tag this cytidine deaminase, you can change a C to a U by this chemical modification. And then when you have this U in the other strand, and that's a mismatch here, the cell would automatically use its mismatch repair mechanism to change this particular G to C combination to an A and T combination. So there you go. You have a G to A change done. Fantastic. So all they had used here is the Cas9's ability to target to a particular area by providing the guide RNAs for that particular area. So that's one of the very first changes that people did or modifications people did to change the base. So that's what they're called base editors. Unfortunately, the base editors only work from G to A, and this one. There's no A to G substitution. So the as I mentioned, the cytidine. So how you get basically from cytosine to uracil is basically take off the amine group at this end, and then you have your targeting Cas9 to a particular region, and you then change the cytosine to uracil. The cell then repairs it to A and T. But what about changing A to G? Well, that doesn't exist because there is no natural DNA adenine deaminase. You have a cytidine deaminase, but not adenine. So, oops. Okay. Oh, God. Sorry, guys. Let's get back. Okay. So what people did, but there is something called the RNA deaminase. So people started engineering by directed evolution. So this is another approach of protein engineering called directed evolution. So they started from a RNA deaminase, which uses RNA as its template, and then changed it to a DNA deaminase, completely artificial, generated in the lab only. But this actually increased the efficiency of generating single nucleotide polymorphism from zero point 0.5 to 1% to more than 50%. So that is a huge, like tenfold, hundredfold jump in efficiency. So this is fantastic news. And this is all done in the lab by using protein engineering. The next thing people did was something called as a prime editor. The prime editor is basically something that works like find and change, something that you already have in Microsoft Word. If you want to change certain letters, you just type in those letters and the software will automatically change that wherever they're present. So this is basically the same thing. So you have it's a bit complex procedure. Uh, I can give you links to it if you want to sort of look at it later on. But at the simplest thing is what they did was they had the Cas9 protein, which is, by the way, it's been sort of deactivated. It only can attach to DNA but not cleave it. And then they have also attached a reverse transcriptase with this one. And then they have something called a PEG RNA, which is a modified guide RNA. And so they come to the DNA, they make a single nucleotide, single strand nick, not a double strand break, but a single strand nick. And then the reverse transcriptase copies the edited sequence, which is present in the guide RNA, to make a new DNA segment. And what that happens is then there's the entire cellular endonuclease will chop off the excess bit that is coming on from the template. And then you have a mismatch here because it has wild type as well as new DNA, and they won't match because they are not exactly similar. And there comes another guide RNA, another Cas9 protein, which nicks on the non-modified strand. And this actually helps or pushes the cell towards repair mechanism, and it uses this changed template as its changed strand, mutated strand as its template and then you have the entire thing completely changed. It's a much more complicated way of doing things, but it works and it's really efficient, and you don't have to, and it doesn't generate any insertion, deletions, or off-target things. So this is a much more, it's, I think this prime editing is not more than 2 years old. So people are, it's still complicated and people are still starting to understand how it happens. But this is how the basic way it happens. Okay, so the last bit, I want to touch upon the different Cas9 proteins because this is the central protein of the process. So people have, of course, made into these kind of different things. There are, which we mostly use is called Streptococcus pyogenes Cas9. That's the wild type one that we mostly use. But there are high fidelity ones which have no off-target effects, reduces it. They have catalytically dead Cas9, which is used for imaging purposes. We have Cas-X and Y, which are very compact, which can be easily packed into cells, easy for sort of, I would say, uh, generation of the Cas proteins inside the cell. Then you have, so Cas9, the wild type, works basically on GC-rich areas, but this Cas12A, it works with AT-rich regions as well. So there's no problem if you have an AT-rich segment. And also it doesn't need a tracer RNA. So there are lots of advantages of having this particular one rather than the Cas9. Depends upon your question and your experiment. Then you have the X-Cas9, which absolutely has got rid of the PAM sequence specificity completely. So you can actually go and do your CRISPR anywhere you want. And then the newest, I think in 2017, they have come up with something called RNA editors. Now you don't have to bother about DNA. You can directly go and change the RNA, which was not done before. And you have this Cas13A and B for as your RNA editors. So these are the upcoming things that you should be looking forward to. There are lots of other things as well I have not put down for the want of time, but this is the sort of broadly the things that you are looking for. And that brings to sort of the last slide, which is the acknowledgement slide, where mean, this is my journey has only be possible because my parents had been extremely cooperative and has made my life a lot easier by not questioning my choices. And then my teachers, all through my life, my bachelor's, master's, PhD, everybody had been very helpful, sometimes pushy, but very helpful in sort of shaping my understanding and my journey. My PhD guide, Dr. Dibendra Sarkar, was extremely good at focusing my mind onto the research questions. Dr. James Locke was where I worked for the initial few years. Very kind-hearted man, let me do whatever I wanted. And currently, my current boss is Professor Claire Bryant, who has actually helped me to develop my CRISPR-Cas9 technology in her lab, and I should really thank her for that. Because I made a transition from bacterial genetics and microbiology to the eukaryotic system. So, and she gave me the opportunity to do that. And these are the funding agencies which have funded me throughout my academic career. And yeah, thank you very much. And if anybody has any questions, I'm more than happy to answer them now. Thank you.
Thank you, Dr. Rajat. There are a couple of questions. So the first question I would like to ask you, yeah.
Okay, ma'am, I'm stopping. Ma'am, I'm stopping sharing the screen now so that I can.
Yes.
Yeah.
Yeah. This is a question posted by Amrita Das from Oxford College of Science. She wanted to know how far this CRISPR technology is effective in cancer therapy.
In cancer. Yeah. Cancer therapeutics. Yeah. Well, it is. So as I mentioned, and as you have already found out, that CRISPR is basically a tool. It's not a whole process. It's a tool to generate certain things easily. And in cancer therapeutics, the most important thing is the CAR T cell therapy, or you have your immune cells, your own immune cells, which had been engineered in a way to fight and to fight the cancerous tumors. So CRISPR-Cas9 gives you a very wide handle as to how easily you can generate those CAR T cell lines. And previously, it used to take a lot of time an effort to generate the mutated CAR T cells, but now it's very easy. So yeah, as a means, this CRISPR will be sort of extremely efficient in generating those CAR T cells. And cell therapy, gene therapy, mm, you can basically have the CRISPR uh, system delivering certain kinds of toxins to your genome, to an area of interest. You can deactivate certain genes in the tumor microenvironment. You can target multiple genes in the tumor in the tumor. So yeah, I mean you can generate lots of options from this, and that's where it will be extremely efficient. I think there are lots of trials going on for different CRISPR-based CRISPR-generated cell lines for cancer therapeutics. I hope this answers your question.
Yeah. Thank you. And there is another question. She's from, I think, UG student, undergraduate student. She wanted to
Yeah.
pursue her genetic engineering further. And meanwhile, she wanted to join some projects, might be is in the form of interns. So is there is any opportunities to get into this field?
Yes. So unfortunately, I'm a little far away from Indian scenario right now, but I guess there are companies in Bangalore itself, which are you have to do a little bit of search, who are actually providing this CRISPR-Cas9 as a service to the many labs, the many like we have this Indian Institute of Science in Bangalore. So there are scientists who want to do this CRISPR-Cas9, but they do not have the expertise in-house. So they outsource these experiments to certain commercial companies. And these commercial companies will, of course, need people to work on these particular processes. And you don't need to be a PhD scientist to do all this kind of steps. There are lots of steps which needs people who are good at their jobs and good at handling cells and good at transfecting and all sorts of stuff. So as an undergraduate, you can probably get into these kind of positions and then build your understanding. You can be inquisitive, you can build your understanding of the system, get some experience, which is absolutely important in industry. And then if you're interested in the system, then you can think of pursuing a PhD or a higher your higher academic career in this particular field. But there are options. There are companies which are sort of willing to take on interns in India. So you can develop your skills that way.
Thank you. Another question is that this is posted by Muhammad Salman Khan. He wanted to know what is the pyroptotic pathway performed in apoptosis.
Well, so just to be clear, apoptosis is a particular type of cell death. Pyroptosis is a completely different type of cell death, so they don't happen together. Apoptosis is different, pyroptosis is different. So the pyroptosis is basically a method where the cell explodes, um, and then so it's not a so apoptosis is basically a silent cell death method where the internals of the cells are eaten up by the tissues, by the cells itself. Autophagy is the outcome of apoptosis at the end. But pyroptosis is not silent. It's extremely violent. It explodes the cell. So the reason it explodes is basically an influx of water into the cell. It bursts the cell and it sort of spills everything that is there inside the cell. And this is also important. Apoptosis does not make an alarm call. Like it's not an immune alarm call process because apoptosis is needed for normal body regeneration, normal tissue formation, morphogenesis. But pyroptosis is basically used for raising alarm for infections, viral and bacterial. So they are completely two different processes of cell death required for two different purposes.
Another person, Guru Raj, has posted a question. Is a CRISPR technology is a license free in case it is used for commercial entities?
Very good question. Very good question. Well, this is a patented technology. No, because people who have done this, developed this, saw the potential in that. It's a patented thingy, but it is free for academic research and non-profit research. This is free. You can get it for free. But if you are a company who wants to make profit by using this technology, then you have to pay a licensing fee to the holder of the patent, and which is in US. So there are a couple of companies who actually sell these licenses to different entities. If you're interested, you pay a fee, and then you are given the authority to use these technologies.
This is a question from Dr. Kavita. How to design guide RNA? Which software to use?
Yeah, another good question. Well, I did not go into the details during my talk, but there are plenty. There are, if you search like how to design guide RNA on Google, I'm pretty sure it will come up like at least 10 different softwares are present. The one I personally use is something called a CRISPR and ChopChop. So these are two web-based services which are developed by academics, like scientists, by studying all sorts of like different combinations of guide RNAs, which are good, which are bad, which are off-target events, how efficient they are, and things like that. So there are quite a few softwares out there, but one needs to take care about which one to use, because every software comes with a manual. So one needs to read those and understand what are the areas where it can be used. Not every software is used for everything. So I think the first steps, if somebody is interested, then they can certainly contact me or somebody else who is an expert in that, and then we can guide them through how to design guide RNA, because that is the most important step of the whole process.
Okay, thank you. This is question from Srinivas. What are the prime opportunities which is available in India and around the world? Which, that is what the question which is posted by him. I hope you understood the question.
Can you repeat it? Just
for the research. Research
okay.
in genetic engineering.
Okay. So, well, genetic engineering comes with a package, right? It's not a single thing that anybody will be interested in, but there are lots of labs in India as well as in around the world who are doing really good science using this kind of cutting edge technologies. So if you're interested in genetic engineering, then and if you're interested to study abroad, then I would probably tell you to focus your attention to the top 50 universities of the world. And how do you know what's the top 50 universities? There's a good university ranking system that comes up. Times Group produces a good list of universities. And then basically you have to spend some time in trying to find a lab which actually you like. And then, of course, apply and try to find if they're willing to take you on. But in India, right now, the situation around the world is quite difficult. Getting a position outside India is quite difficult and it's complex. So, but in India, there are lots of young labs, people who have come back from abroad. They're also they're bringing back this kind of technologies to India and using them. So I would suggest you go for the national sort of the scholarship program of Indian government, which is quite generous, and then try to sort of develop your research career inside India as well. There are good opportunities there.
We can take few more questions. Sure.
Yep.
Okay. Is there any replacement for Cas9 protein?
Cas9 is sort of a generic protein that was first isolated from, I think, Streptococcus pyogenes, and that has been widely used. But there are multiple Cas9 proteins in many prokaryotes, in many bacteria. We even don't know many of them. So one of the active fields of research in many labs around the world is to try to find out replacement for Cas9, better replacement for Cas9. I have highlighted a few. Some of them are naturally occurring in different species of bacteria. So there's I think two particular labs which sort of dedicate their attention in finding out different Cas9 molecules from bacterial genomes as well as archaeal genomes, and they have been extremely successful. So yes, there are replacements. Some of them are already in use, but you can of course do more prospecting as well.
Another question, again from Soumya, is there is any difference between the pyroptosis and necrosis, or both are same? Because both of it affect the nearby cells. The
Good
difference
question.
between the pyroptosis and necrosis.
Yes. So the pyroptosis and necroptosis are very similar processes, except that it uses completely different molecular pathways to do the same thing. And necroptosis is sort of the old, in evolutionary terms, it precedes the pyroptotic process. Because necroptosis you will find in pretty much all species, all different phyla of the vertebrate sort of family. But pyroptosis is sort of limited to a certain uh, sort of subclass of vertebrates. And also, even if they're present in certain animals, they are not used in mammals for infection fighting. They are necroptosis is mostly used for viral methods to stop viral infection. Uh, so paroptosis is also used for it's mostly used for bacterial infection detection and to rectify that. But they're very similar processes, similar outcomes, but the starting material, the starting point, or the signal that activates pyroptosis and necroptosis are quite different.
This is posted by Juhi from the Oxford College of Science. She's pursuing her MSc in biotechnology right now. She wanted to know, after completing the MSc biotechnology, is that a satisfactory for the work or the research, or she wanted to take any other online or offline courses to get into any research?
Okay, so there are two types of research that you can actually go into after your master's. One is industry based, and the other one is sort of through the academic world. If you want to join a company, then masters in biotechnology, as taught in India, will be sufficient to try to join different industries. But there are lots of hands-on courses. So online courses are not very helpful in life science research, because at the end of the day, you have to actually go into a lab and do hands-on work. You have to hands-on experience. So you have to have some kind of a hands-on training on different aspects which are not broadly taught in biotechnology course. Uh, so there are certain companies in Bangalore back then, even I think they have developed even now. So they provide like a couple of weeks or a month's course where they teach you molecular biology, more advanced molecular biology techniques, protein purification techniques, and things like that. So if you can do that and then join the industry, that will be, because that's a good way of getting into industry as well. The other way, and I have my friends from my batch in Oxford College, they are working in R&D without doing a PhD. They have done the masters from Oxford, and they have joined companies in Bangalore, in Pune, in Hyderabad, and they are working as R&D specialists, scientists, without doing a PhD. So the other one is basically through academic light, which is sort of trying to get to the industry, but gaining a PhD in the medium term. So you can go through both the ways. If you are not interested in sort of doing a PhD, you don't have to do it. It's not a compulsory. Masters should be good enough, but do some hands-on courses to improve your skill set for the industry, because industry doesn't care. They want good people to work with.
This will be the last question, I think. Is there any disadvantages or the drawbacks of CRISPR technology?
Yes, of course, as with all technologies. So the biggest drawback of CRISPR is something called an off-target event. Like you sort of tell the the Cas9 protein to go and bind somewhere in the genome, and you expect them to bind only there and nowhere else. But the problem is these off-target events, because it's a 20 nucleotide sequence and the genome is so many megabases, that particular combination will, of course, be found in some other places. Maybe there will be a couple of mismatches, but there will be still substantial match where it can go and bind. And wherever it can bind, it will cut. So there are strategies and there are methods which minimize the whole possibility of an off-target event. But as of now, there is no sure shot way of eliminating this particular problem. So that's why you need to screen a lot more. You have to design your experiments carefully so that you don't have a false positive in your experiment. So the more deeper you go into it, there are lots of other strategies that scientists use to sort of get around this problem. But this is the biggest problem that CRISPR faces as of now.
So, is there
Great.
is any field where exactly the CRISPR has been applied?
Oh, it's been applied everywhere. It's actually so if you talk about basic research, it's everywhere. Every single lab who wants to delete a gene uses this now, and the animal work is being discouraged, so you don't get transgenic mice that easily. So the alternative is basically having a cell line where you have deleted your gene. It's faster. So every lab sort of tries to do that. The other field is, of course, therapeutics. So the CRISPR has been used to treat HIV infections. It has been treated single gene changes, like as I mentioned, sickle cell anemia. There's I think a clinical trial going on for treating sickle cell anemic patients with CRISPR, so in vivo, inside the human body. So that's the ultimate step people want to go for. As I mentioned, cancer therapeutics, you can sort of make different cell lines pretty easily with this. The diagnostics, there are certain Cas9 proteins which are very useful in diagnostics, and also I think there's a company already making diagnostic kits for COVID-19 detection in the US using CRISPR. I think some of them also been tested in the UK, but so these are the few fields where they have already been used extensively.
Okay. She also wanted to know, is this CRISPR technology is not being used in any particular field? It
Well,
has
where there's
not been
no
applied.
hard to say. As I mentioned, if you want something to be deleted, doesn't matter which species you have, you use it. So there's not any particular field I know of where it's been actively discouraged, actively not used. You can even use that for plant modifications, for plant biotechnology, for means means everywhere. But CRISPR is not used in bacteria because it's already present in bacteria. You can't really use them inside the bacteria to do genetic deletions. So that's one of the issues to keep in mind. You can use that in eukaryotes, but not in bacteria.
Okay. There are so many questions which is still going on. I mean, I'm getting. So can we proceed with it, or it is a time actually you're taking long?
I can take so I'm available till another, let's say, 15 minutes.
Okay.
Then I have to go.
Then fine. We can take another 2 more questions. Okay.
Yeah, sure.
Yeah. This is the question. Are the gRNA and Cas9 not degraded by RNAs and protease respectively when it is delivered into cell?
Very good question. Whoever did that, very good question. Yes, they are degraded, and that's why you need a constant supply inside the cell. So if you have a plasmid-based delivery system, then the plasmid generates the guide RNA and the protein continuously, constitutively inside the cell. That's how you get rid of this problem. The other one is if you use purified protein, then you have to do this CRISPR electroporation multiple times to give that more time to do its job. But yes, they are degraded inside the cell.
Okay. So this is the last question. To take care of off-target tissue, do we need to design more than one oligo for a gene?
Very good question. Very, very insightful. Yes. So the problem is if you have more than one oligos in the same cell, then you are going to have problems and if you're targeting the same gene, because they might collaborate together and sort of delete a portion and then repair it in a way that you don't want. So it's not plain and simple using two guide RNAs for the same gene at the same time. But there are strategies which use two guide RNAs, and I'll give you an example how it works. So Cas9 generally makes a double-stranded break, but Cas9 has been modified to produce only a nick in only one strand of the DNA and not a double-strand break. Now you can use one guide RNA to nick one strand and then use another guide RNA to nick the other strand. And this happening in multiple places, the probability of this happening in multiple places in the genome is quite low, because you have to have the two guide RNAs doing exactly the same stuff somewhere else where you don't want it to. And they can of course individually do somewhere else, but that will be of no concern because it will not create a double strand break. So this way you can sort of get around by the issue of off-target events by using two guide RNAs. But that's a very smart question.
Thank you. So thank you all for this wonderful interactive session. And it's my privilege to propose vote of thanks to our eminent resource person and again our proud alumni for accepting our invitation and volunteered to spend his time and share his knowledge with us. Thank you, Dr. Arijat, and we look forward to your
support pleasure.
And continuous relationship among TBOPS. I would also
Absolutely.
like to
Thank you.
Thank you. I would also like to thank our chairman and principal for their guidance and valuable suggestions for the smooth flow of the event. I would like to convey my deep appreciation to my fellow colleagues for their contributions toward the smooth conduct of the event. Last but not the least, I would like to thank all the participants from various colleges, universities, and research institutions from all over the country and abroad for their presence and participation. Thank you all. Knowledge is complete only if it is garnished with the human feelings of compassion, tolerance, discipline, and devotion. With these words, I would like to end day 2 session. And this is Mamta, Associate Professor, Department of Biotechnology, signing off for the day. And see you all tomorrow again with a day 3 session on scope of environmental biotechnology. Thank you all once again.
Thank you. Take care. Bye.
Thank you so much.